Hi everyone, welcome back to our molecular biology series. Today we are learning about one of the most ambitious scientific projects in human history, the Human Genome Project. Imagine trying to read all 3 billion letters of the human DNA code. That's exactly what scientists did between 1990 and 2003. This project revolutionized biology and medicine. Let's understand what it was, how they did it, and why it matters. Here is the question that sparked everything. Why are people different? You and I look different. We might get different diseases. We respond differently to medicines. But why? The answer lies in our DNA sequences. If two people differ, their DNA sequences must differ, at least at some places. So, scientists thought, what if we could read the entire human DNA sequence? What if we knew every single gene, every letter of the genetic code? That would be the ultimate instruction manual for building a human being. This ambitious idea led to the Human Genome Project. To appreciate the project's magnitude, consider its staggering statistics. The human genome contains roughly 3 billion base pairs. That's massive, right? With an estimated sequencing cost of $3 per base pair, the total projected price tag reached approximately $9 billion. Dollars. That's a huge budget. Another way of estimating this is if you printed one person's entire DNA sequence, it would fill about 3,300 books, each consisting of 1,000 pages and 1,000 characters per page. This was truly a mega project that demanded unprecedented levels of funding, advanced computing, and global teamwork. This project had six major goals. First, identify all human genes. They estimated around 20,000 to 25,000 genes. Second, determine the sequence of all 3 billion chemical base pairs that make up human DNA. Third, store massive this massive amount of information in database that scientists worldwide could access. Fourth, develop better tools to analyze this data. This led to a whole new field called bioinformatics, combining biology with computer sciences. Fifth, transfer these technologies to industries and other sectors for practical applications. Last and very important, address the ethical, legal, and social issues. What about genetic privacy? Could this information be used to discriminate against people? These weren't just scientific questions, they were human questions. Launched in 1990 and completed in 2003, the Human Genome Project was a 13-year international collaboration led by the US Department of Energy and National Institute of Health, with major support from United Kingdom's Wellcome Trust, involving contributions from nations like Japan, France, Germany, and China. The project stands as a landmark achievement in global scientific cooperation. So, now the question is, how did they do that? They used two major approaches. Approach 1, gene focused, and uh, approach 2, shotgun sequencing. Scientists used two major strategies. The first approach focused on identifying genes that are actually expressed, genes that make RNA and proteins. These are called expressed sequence tags or ESTs. This was the smart, targeted approach. Find what's functional first. The second approach was far more brute force. Just sequence everything, every bit of DNA, whether it codes for protein or not, whether it's genes or junk DNA sequence, just sequence it all. Then later go back and figure out which parts do what. This is called sequence annotation, like taking notes on what each region of the DNA actually does. Both approaches were used, and combining them gave us the complete picture. Here is how this actual sequencing worked. First, extract all the DNA from human cells. Second, break this DNA into smaller fragments. If you remember, DNA is incredibly long, and the technology couldn't sequence very long pieces all at once. The next step was to clone these fragments in bacteria or yeast using special vectors called bacterial artificial chromosomes and yeast artificial chromosomes. Cloning amplifies each fragment, making millions of copies, so there is enough to work with. Next, sequence these fragments using automated DNA sequencers that worked on the Sanger method. Fifth, and this is tricky, assemble all these sequences by finding overlapping regions between fragments. It's like putting together a 3 billion piece jigsaw puzzle. So, how does assembling the sequences using overlapping regions worked? Let's try to understand it first. Think of it like reassembling a shredded letter by finding matching words. If in our first example, the fragment A says the quick brown, and fragment B says brown fox jumps. So, if fragment A ends with brown and fragment B starts with brown, you know they overlap and connect. By layering different shreds of the same message, you can align these matching segments to bridge the gap and reconstruct the full sentence. So, because brown appears in both, you know that fragment B must come immediately after fragment A. And so the assembly would be like the quick brown fox jumps. And in the other example, try to think again like a shredded letter that you're trying to tape back together. If you have two copies of the same letter and shred them differently, you can find the overlaps to rebuild the message. So, fragment 1 is meet at the park at, then the park at noon on, and the fragment 3 is noon on Tuesday. So, the assembly would be like uh, by matching the bolded overlaps, you can see exactly how the fragments plug into each other. So, the assembly would be like meet at the park at noon on Tuesday. This required specialized computer programs because humans couldn't possibly do this alignment. Finally, annotate the sequence, figure out which part are genes, which chromosomes they belong to, what they might do. The last chromosome to be completely sequenced was chromosome 1, finished in May 2006. The project revealed some surprising things. The human genome has approximately 3,164 million base pairs, close to the 3 billion estimate. The average gene is about 3,000 bases long, but sizes vary widely. The largest known human gene is dystrophin at 2.4 million bases. And here is the shocker. We only have about 30,000 genes. Scientists had predicted 80,000 or even 1,040 lakh 40,000 genes, but the actual number is much lower. Humans aren't as genetically complex as we thought. Another beautiful finding, all humans are 99.9% identical at the DNA level. We are far more similar than different. But here is what keeps scientists humble. We still don't know what over 50% of discovered genes actually do. Half of our genome is still a mystery. Here are more findings. Less than 2% of the human genome actually codes for proteins. Just 2%. So, what's the other 90%, 98% is doing? Much of it is repetitive sequence, stretches of DNA that repeat hundreds or even thousands of times. Scientists initially called this junk DNA, but we are learning it has roles in chromosome regulation and structure. Chromosome 1 has the most genes, 2968, while the Y chromosome has the fewest, just 231 genes. And scientists identified about 1.4 million locations where single bases differ between people. These are called SNPs, which stands for single nucleotide polymorphisms. These small differences are what make us unique. They affect disease risk, drug response, and physical traits. Why do why did we do all this? What's the point? The project's applications are revolutionary, particularly in personalized medicine, where DNA insights allow for tailoring treatments and identifying genes linked to diseases like cancers and Alzheimer's. Beyond healthcare and medicine, sequencing various organisms from bacteria to crops advances agriculture, environmental cleanup, and our understanding of human evolution. The Human Genome Project changed how we do biology. In the past, researchers studied one or a few genes at a time, slow and limited. Now, with the complete genome sequence and new technologies, we can study all genes at once. We can look at how tens of thousands of genes and proteins work together in a complex network to create life. This sequence is just the beginning. The real challenge is turning this raw data into meaningful biological understanding. What does each gene do? How do they interact? How do mutations cause diseases? These questions will keep scientists busy for decades. We have the instruction manual, now we need to truly understand the language. So, here is the summary. From 1990 to 2003, the Human Genome Project was a massive international effort that sequenced all 3 billion DNA base pairs and identified roughly 20,000 to 25,000 genes. Key findings revealed that humans are 99.9% genetically identical, and about 1.5 to 2% of our DNA actually codes for proteins. This work changed medicine through better diagnostics and personalized treatments, while also giving rise to bioinformatics, which is the integration of biology and computer science. Research continues today as scientists further analyze this fundamental data. So, here are neat must know facts. Master these key Human Genome Project facts that are highly yield topics frequently tested in NEET biology. If this helped you understand the Human Genome Project, please stay here and subscribe for more molecular biology videos. The next video will be DNA fingerprinting, how we use DNA to identify people. See you there.